Yeah, so thank you very much for everyone and for your help as well setting up everything. And thank you so much for the Neurological Foundation for the opportunity of presenting our research, what we're doing in the lab at the moment. And I will focus on the gut-brain axis in Parkinson's disease, but I wanted also really to focus on neuroinflammation and how healthy lifestyle and well-being could reduce it, neuroinflammation, and how it can have an impact that maybe could slow down or even prevent some neurological disease, such as Parkinson's disease, but also like other psychiatric disease and neurodegenerative disease in general. It doesn't want to switch. Yep, there we go. So I'm just going to explain a bit my journey. So I'm from Belgium. This is a nice little country in the middle of Europe. We have neighbors with the Netherlands, with Germany, with Luxembourg, with France. I think I might have some friends watching from France. So hi. We have different languages in Belgium. So the Dutch part, it's a flatland in the Dutch part. I put some Maori name as well because I like their way of introducing where we're from. So Papeete, it's flatland. We have a sea, Moana. We have a French part of the country, so you can guess it's where I'm from, where we have more hills like Puke, and we have a German part close to German border. And we're very famous for the waffles, for nice buildings, for French fries. Well, actually, they're not French, they're Belgians. And also for beers, they're delicious. So my studies, so I did a bachelor degree in biomedical science. There we go. Sorry, I'm just gonna check this here. So I did a bachelor degree in biomedical science and then a master degree in immunology and oncology. And then I did a PhD. The slide doesn't want to keep going. Yeah, there we go. Then I did a PhD on neuroinflammation and neurotropic viruses. And I did that PhD in Brussels at the Scientific Institute of Public Health, which is here. And also in collaboration with the University of Ghent and the VAB Center for Inflammation Research. So it was a collaboration between two institutes. And that was very lucky for me because I had some experience from virologists and experience from people in inflammation. So my previous studies and project, it's important just to understand, the global research we're doing. So I did my master degree in inflammatory microenvironment of the lung and how that microenvironment of the lung could promote metastasis from a distant tumor in mice. So for that, we were inducing inflammation in the lung, and inflammation in the lung can be induced by smoking cigarette, for example, or people with asthma. And we discovered that yes, that inflammatory environment could increase the progression of metastasis within the lung from a distant tumor, like, for example, a subcutaneous tumor. And then during my PhD, I studied a gene called MALT1, and that gene was involved in NF-kappa B activation. And NF-kappa B activation, it's a transcription factor involved in inflammation. And I wanted to see if inhibition of neuroinflammation could decrease pathology and pathogenicity of a rabies virus. And for that, I was using different virus strain. And I could see that neuroinflammation was important by clearing a virus from the brain. When I was using an attenuated type of virus, such as virus were used for vaccine, for example. But it was also very important in inducing pathology when I was using a rabies, a virulent, very pathogenic rabies virus. So I discovered that neuroinflammation could be beneficial and detrimental at the same time. And here, the connection of those projects were really the impact of inflammation in disease. It can be protective, but can be detrimental again. Important for me, it's to explain that I took some break from science. After my master degree, I went to Australia for a year. I was working there in different area than science. And it was to be away from a stressful year during my master, going back to meaningful social interaction, be more in nature, just really focus on myself and my well-being. And after the PhD, I went to Canada for a year as well. Same, just to go back to social interaction after stressful PhD, focus on myself to be more healthy, which I was already adapting that in my PhD, hiking, being in nature, meaningful social interaction. Especially met one of my best friends now, Delphine. She's on that picture. I just didn't put her face because I don't think she would like to be there. But she helped me. She helped me to understand that actually she was dealing with some stress, anxiety, and depression. And her way of treating that was really to be in nature and to hike as well. So it was very triggering for me to understand that better, saying, okay, that's actually a good way. And it just like helped me to be more interested in that in the future. Then I went, after Canada, I went to New Zealand, where I joined Louise Parbrunley's lab. And the purpose on that, so no, first I was working as a barista in Picton, and then I applied for a job at Louise Parbrunley's lab as an assistant research fellow. And the idea here was to work on what she was already working on optogenetics, which is an alternative to deep brain stimulation to treat Parkinson's disease when it's at a late stage of the disease. And I wanted also to connect the dots of everything I was doing before. And Louise allowed me to write my review and just to combine what I was doing before and what I was doing at the moment working on Parkinson's disease. And one of the hallmarks of the disease, of Parkinson's disease, is in neuroinflammation. So I could work a bit on that, but I also wanted to be a bit more broad and include my research on immunology, the importance of well-being, physical and mental and healthy lifestyle through my travels, research on neuroinflammation, as I explained, and also like dealing personally with some gut problems already to be healthy. And all of that, so again, Louise allowed me to write my own review, so that was very interesting for me. And also it was when the first COVID lockdown happened, so I could really focus on the writing, which was a good timing for me. And all those dots connect together, linked to the immune system in the body and in the brain and in diseases in general again. And again, as I explained, it can be beneficial or pathogenic. So I'm going to focus more on neuroinflammation just so everyone understands better what is it. Here it's a very simplified schema, just explaining those different cell types involved. And there are even more than that, just to have a better idea in general. So neuroinflammation occur in the brain and involve neuronal cells in the brain. They can, when they're damaged by a stimuli, by a pathological event, they can secrete some factors that can activate immune cells, resident immune cells in the brain. Those cells are the macroglial cells and astrocytes. So those cells normally in the brain, they're protective and they will help to just get rid of toxin in the brain or just damaged cells that are not needed there anymore. And astrocytes are very protective for the brain as well by bringing nutrients and just consolidating the brain structure as well. So they're very important just in a healthy brain in general. But when there's that pathological event, they will start to secrete some mediator. So they are called pro-inflammatory factors, such as pro-inflammatory cytokines and chemokines. And also they can recruit innate immune cells that are not normally present in the brain, such as dendritic cells, macrophages, natural killer cells. All of those cells are normally not specific from the pathogenic event. So they're just the first defense line against a pathogenic event. But then they can also secrete those pro-inflammatory cytokines, leading to the recruitment of cells specific from the pathogenic events, such as B cells and T cells. So that process normally, when it happens just very acutely, it can help to decrease those toxins from the brain or viruses from the brain. And it's normally a healthy process to combat disease. But when it's chronic, it can be destructive for the neuronal cells within the brain. And this is a more bigger schema, just explaining just in a better way, just so you better understand those concepts of barrier within the brain and what's happening in the blood, what's happening in the brain. So you will better understand what's happening later on in my presentation as well. So normally there is a stress or trauma infection pathology. We'll activate those brain immune cells, resident cells in the brain. So they will secrete pro-inflammatory factor, which further can recruit some innate immune cells from the periphery. So when I say periphery, it's from the blood. They normally don't invade the brain, and there's that blood-brain barrier that's called. So this is that barrier here. But so yeah, when there's a pathologic event, they can migrate into the brain and cause neuroinflammation because they will secrete pro-inflammatory cytokines and chemokines. Further on, they can also lead to the recruitment of those cells from the adaptive immunity, specific from the pathogenic event, which will lead further to furthermore even neuroinflammation. That process will lead then to damaged neuron, leading to neurological disease because those neuronal cells will die. Those cells, by dying, will secrete even more factors that can lead even more to the activation of those microglial cells and astrocytes. This means that it's a vicious circle, mean that it just keep going, and it's become chronic, and that's when it causes the pathology. There's different way of measuring neuroinflammation and to analyze it. We can do that in animal models and in human patients. I will focus a bit more on animal models at the end of that presentation, but in human patient, so it's still very simplified as well. It's even more technical than that, but just so you have a good idea. So we can measure, for example, the level of pro-inflammatory markers in the blood, like C-reactive protein or pro-inflammatory cytokine and chemokine, but they're not directly linked to what happened in the brain. There's still inflammation in the blood, means that might be inflammation somewhere else in the body, not necessarily in the brain. But in the brain, we can also use those imaging techniques like the magnetic resonance imaging, MRI, to detect structural changes associated with inflammation. And we can also use the technique called positron emission tomography or PET scan. This will allow to analyze specific cells or compounds in the brain. For example, here we can use a specific marker of microglial cells activation. So this is very helpful to understand if there is neuroinflammation during certain disease in patients. We can also post-mortem, so when the patient has died, we can do brain immunohistochemistry by analyzing all those cell types involved in the brain and if neuroinflammation was present. And we can also analyze the cerebrospinal fluid or CSF, analyze also those pro-inflammatory markers, and they are better representation of what's happening in the brain. So during my review, it was very interesting on what's happening in human patients. There are many studies that have done that. So microglial cell activation via PET scan in human patients, and could see that actually neuroinflammation is present in a lot of different diseases. So neurodegenerative and psychiatric as well. So Huntington disease, multiple sclerosis, Parkinson disease, bipolar disorder, Alzheimer's disease, depression, schizophrenia, and amyotrophic lateral sclerosis. And very interestingly, when we correlate that neuroinflammation with cell death in neurodegenerative disease, we know that neurodegenerative disease, one of the hallmark is the cell death in different brain regions. The region will depend on the disease. And I wanted to see like if those neuroinflammation will correlate to the region of cell death in neurodegenerative disease. So analyze that as well in different studies. And we could see that actually, yes, like for Huntington disease, Parkinson disease, Alzheimer, and amyotrophic lateral sclerosis, they correlate, microglial cell activation correlate to the cell death region for those diseases. So now I'm going to focus on, so this was just to explain that actually neuroinflammation could be, a cause of those cellular death in the disease, but it could also be a consequence. So we still don't know that, or it might be both. So here I'm just going to focus on Parkinson disease because it's what we work in the lab at the moment, and we're going to explain a bit better what's the disease. So Parkinson disease is the second most common neurodegenerative disease in the world after Alzheimer's disease, and it's due to the death of brainstem dopamine neurons. So the brainstem is just situated here, as you can see here. And normally, this is a healthy brainstem, healthy substantia nigra full of dopaminergic neurons. In a patient with Parkinson disease, there is a diminished substantia nigra. You can just see it by the eye, just like that. And because all those dopaminergic cells, almost all of them are dead. Another hallmark of the disease is the accumulation of vesicles called Lewy bodies. Those Lewy bodies have some aggregates of a protein called alpha-synuclein. Normally, that protein is present in the body, in the human body, but they don't aggregate and they don't phosphorylate here. They just aggregate together and they're pathological in Parkinson's disease. Very interestingly, and what we have to explain here is that after it takes many years for the dopaminergic cells to die. And there's that window of here, like the patient will only present the motor symptoms when there's almost like 50% of the dopaminergic cells that have died. So the loss of dopaminergic cells means loss of dopamine. Dopamine is a neurotransmitter in the brain that is involved in reward, in motivation, in pleasure as well. But it's also involved in movement. So because we have less cell producing that neurotransmitter, we have less dopamine. It's involved in a circuit called the basal ganglia thalamocortical circuit, which when we have a decrease of dopamine, that whole circuit is impaired, leading to decrease of movement. And that's why we observe all those impaired movement in Parkinsonian patients, such as tremor, postural instability, rigidity, tremor, shuffling step, etc. Those patients also present non-motor symptoms such as cognitive dysfunction, also autonomic dysfunction, problem with sleep as well. And yeah, so the different cause of the disease, they're aging and genetics. Normally we can modify those causes. Genetics, it's not present in all of those Parkinson disease patients. It's like around 5 to 10%, if I remember well. But there's also that component of the lifestyle and environmental factors where we can actually act on those factors. Interestingly, the motor symptoms just show at the late life when it's the motor phase. And I explained before that during that stage, almost most of the dopaminergic cell already dead. So we can't really act on that, at least on the dying of those cells. But very interestingly, there's a prodromal phase of the disease during the midlife when we can start to see already clinical biomarkers of the disease. So such as olfactory impairment, rapid eye sleep behavior disorder, gut dysbiosis, autonomic dysfunction, depression and anxiety, and somnolence. So the gut dysbiosis, it's the imbalance between good and bad bacteria in the gut. So this is very important because they're called clinical biomarkers because it's important. If we know that they actually start to have Parkinson's disease at that stage, we can have an impact earlier on in the disease and maybe prevent the death of those dopaminergic cell death. Because at the moment, there are only one treatment, well, a few treatments available only when the motor phase appear, just to help to treat those motor symptoms. And it's called L-DOPA treatment. So it's a dopamine agonist that will be administered to patients. So they respond very well to that treatment for the first year of life, as you can see in that window. But after a few years, around 7 to 10 years, depending on the concentration of levodopa in the system, even if it's too low, they will start to have again those symptoms associated with Parkinson's, such as akinesia and rigidity. But if the concentration of levodopa is too high, they will have a new type of motor symptom, just very linked to that treatment, which is called dyskinesia. And it's a bit of uncontrolled movement, really typical from Parkinson's disease, such as what has Michael J. Fox. For those patients who don't respond well to the treatment, there is a surgery that can be offered if they're fit enough for that. It's called deep brain stimulation, where we actually going to place an electrode within the brain, and it's going to stimulate the area, and it's going to help with the motor symptoms. The problem with that technique is that it stimulates all different cell types in that brain region, and some cell types are excitatory, some inhibitory, and we just don't want to stimulate everything there. So that's why when I arrived in the lab, Louise was working on optogenetics, and I started to work on that as well. And it's a very new technique that can actually stimulate the cells, very specifically those cells that we want without touching other cell type, just by using light and virus vectors. So this is a very interesting new technique. The problem is it's still at the late phase of the disease. And here really, we really want to focus more on what's happening before. And the prodromal phase of the disease really tells us and tell the patient there's actually something already going wrong there. So just wake up, you need to act on something. So this is very important for clinician and just researcher now to have biomarker of that phase. So this is when we could act very early. So as I explained just here, lifestyle and environmental factor could be cause of the disease and are the cause of the disease. So when we analyze a bit more what were the risk and protective factor of Parkinson's disease, there's a lot of research out there, a lot of epidemiological studies and just preclinical studies as well. And this was the result. So we could see that risk factor of Parkinson's disease are trauma, virus infection, gut dysbiosis, so that imbalance between the good and bad bacteria, exposure to pesticides, drug exposure, stress, a huge factor. Those, non-healthy foods, such as food very high in trans and saturated fat, sweet beverage, etc., but also melanoma. And processed meat and red meat, apparently, it's also a risk factor. And those protective risk factors were exercising, just nutritious food rich in veggies and fruits, good fat found in fatty fish and also in avocado. Also, the use of ibuprofen through life as well would be a protective factor for Parkinson's disease. Drinking coffee, drinking tea, the resveratrol present in the red wine as well, moderate consumption. And apparently cigarette smoking would be protective for PD, but it would be probably just one of the components like nicotine. And that's so we definitely don't tell people you should smoke to not have Parkinson's disease, still have a lot of bad health effect, especially on the lungs. So the idea here would be, of course, you can suppress all the risk factor, but it's really to put that balance towards the protective factor and, have from time to time a good McDonald's. Yes, that's all right, but just like try to be healthy most of the time and you would reduce your chance of having Parkinson's disease. And very interestingly, when we analyze if those factors were linked to inflammation, a lot of them actually are linked to increase of neuroinflammation, those risk factors, or decrease in neuroinflammation, so those protective, but not all of them. And very interestingly, all of those factors, when you see it's like food, there's also that exposure to pollution, and all those factors would enter through the mouth and the nose. And there's that hypothesis, which is called the Brake hypothesis, and it's more and more approved right now, that actually Parkinson's disease wouldn't start in the brain itself, but would start in the gut. And why that? So there is that hypothesis that by all those administration of environmental factor, you would induce gut inflammation, but also disbalance between the good and bad bacteria in your gut. That can link to accumulation of that alpha-synuclein. Alpha-synuclein accumulation and aggregates can travel through the vagus nerve to the brain. The vagus nerve is normally linked, the brain and the gut and help in digestion, and also other physiological processes. So the alpha-synuclein could travel the vagus nerve. Inflammation in the gut as well. There's also that inflammation in the olfactory bulbs, which is the entry of pollutants, for example. This inflammation can also lead to accumulation of aggregates, alpha-synuclein aggregates. Inflammation in the gut can also lead to systemic inflammation, going through the blood-brain barrier, causing neuroinflammation in the brain. And all those processes together, so you take the inflammatory bulb inflammation, olfactory bulb inflammation, neuroinflammation, accumulation of alpha-synuclein in the brainstem, all of this would cause neurodegeneration, and then Parkinson's disease. It's neurodegeneration of the dopaminergic cells. So that process would take years. What's very interesting here is that by analyzing different studies as well, we could see that actually inflammation is already present in the prodromal phase, especially olfactory bulb inflammation and the gut inflammation, which is actually, it makes sense because in the clinical biomarkers of the prodromal phase, you can see olfactory impairments and also gut dysbiosis and gut problem. And we know that patient, Parkinson's disease patient, we start to have some gut problems as well. So it means that neuroinflammation starts early and can last, during the whole disease. It means that we can target neuroinflammation at that stage, at the prodromal phase, and maybe by targeting at that stage, it would slow down the disease and slow down the progression. So being a neuroinflammation nerd, I wanted to analyze more what's happening in the brain in Parkinson's disease. For neuroinflammation, it's a huge schema, so I won't explain everything. But what I really want you to focus on is that there are different factors of neuroinflammation, and just focus on those bubbles. So those orange bubbles are detrimental immune factor in Parkinson's disease, and those yellow are protective immune factor in Parkinson's disease. If they're protective in that disease, doesn't mean that they're not protective or they're detrimental in other disease. It's just very linked to the disease. And the green one are those factors where the role is unclear. It's very interesting because a lot of them are protective, a lot of them are destructive, especially activated pro-inflammatory microglia. It's a hallmark of those diseases. We know it's destructive here. Activated astroglia as well, secretion of pro-inflammatory markers, etc. For the protective immune marker, anti-inflammatory stage of microglia can be protective, and astroglia as well. Natural killer cells could help decrease the accumulation of alpha-synuclein in the brain. And there's a new system called, well, it's not a new, it's always been there, but we just discovered very recently that the glymphatic system, it's a system activated when we sleep, and it can help decrease the toxin level in the brain. And that's why sleep is so important, and it could contribute to the decrease of alpha-synuclein aggregation in the brain. But also, we're not sure if it also participates to neuroinflammation and might be this destructive, so it's still unclear at the moment. So the main idea here is there is a lot going on. It's very complex, but there's still some protective factor and some destructive. So when we say, oh, maybe we could use anti-inflammatory drugs in Parkinson's disease, well, it would just decrease everything, but still some factor are protective. So that's still kind of unclear what can be used at the moment. And there's a lot of research about anti-inflammatory approach in Parkinson's disease out there, especially in preclinical models, but also in the clinics. And what's out there is drug already on the market for other disease, such as immunosuppressant, but also ibuprofen, just anti-inflammatory in general. There are drugs targeting transcription pathway involved in inflammation, such as NF-kappa B, like I talked earlier on. Drugs targeting pro-inflammatory cytokines, drugs targeting the gut inflammation and gut microbiota. This is a new era as well, and it's very promising. And drug converting pro-inflammatory microglia to anti-inflammatory. And especially I found that there's a lot of natural anti-inflammatory compounds that could be used to decrease the inflammation stage and neuroinflammation in Parkinson's disease. And maybe that could be the key, because maybe natural anti-inflammatory compound would just help to go back to homeostasis in the brain and go back towards a more healthy state, compared to, an immunosuppressant would just suppress everything. And also we know that those immune cells are important for the normal brain function. So I was very interested also to know that there are anti-inflammatory strategies via lifestyle, and just wider than Parkinson's disease, neuroinflammation is involved in all those diseases again. So I won't repeat them, but yeah, psychiatric and neurodegenerative disease. And just wanted to check in the literature, what can we do to reduce neuroinflammation in general? And I found a lot of studies, a lot of them, and which is very good, because it means that we can have an impact ourselves to decrease that neuroinflammation, and maybe just the fact to develop neurodegenerative and psychiatric disorder in general. The idea would be, so these are all bubbles anti-inflammatory, and those would be inflammatory. And the idea is to act on that during your life. So it's not like doing that 1 week and then it's gonna be all right. No, like you have to act on that during your whole life. And hopefully by increasing those bubbles, you will promote positive aging. But if you do more of those bubbles, you would promote more neurodegenerative and psychiatric disorders. Those bubbles were exercise, healthy diet, nature, ketogenic diet, healthy gut microbiota, yoga, mindfulness, meditation, intermittent fasting. Whereas anti-inflammatory bubbles, were stress, obesity, gut inflammation, gut dysbiosis, pollution, early life stress, pesticide exposure, high-fat diet, heavy metal exposure, and sleep deprivation. So yeah, this is very, very interesting. Yeah. What was very interesting as well, it's when you analyze those bubbles, most of them are linked to the food consumption. So all the diet here and, obesity and everything. So it looks like the gut, there's something between the gut and the brain somehow, and the gut and neuroinflammation as well. Which actually it's already known that there's some anti-inflammatory approach in diet, and where some food are anti-inflammatory, other food are inflammatory. So the anti-inflammatory food would be the tomatoes, olive oil, green leafy veggies, nuts, fatty fish, fruits. Whereas the inflammatory food would be refined carbohydrates, French fries, other fried food, soda and sugar, sweetened beverage, red meat, especially processed meat, margarine and lard. But what's the link with the brain? So there's a huge link with the gut and the brain. So it's called the gut-brain axis, and it involves different systems. So we know that the enteric nervous system has millions of neurons. So the gut is also called a second brain. And the gut itself, the enteric nervous system, is part of the autonomic nervous system. And the autonomic nervous system also includes the vagus nerve. So this will be what's help the digestion in general. And we also know that actually the gut can, especially the bacteria present in the gut, can produce neurotransmitters, which are GABA, serotonin, dopamine, oxytocin, but also other ones. And those would be directly linked to your behavior and your mood, and yeah, the fact that you're depressed or anxious as well, based on what type of neurotransmitter they would secrete as well. So this is very important to have a healthy gut microbiota. And there's also that endocrine system. So they can communicate together via the endocrine system, especially the hypothalamus pituitary adrenal axis, which is involved in the stress response. So when there's a stress, the brain will release some hormones acting directly on the pituitary, hormones again acting on the adrenal glands, releasing some cortisol, which is a substance that will act directly on the gut and can straight away alter the gut barrier and the intestinal barrier as well, which is not surprising. I don't know you, but for me, when I'm stressed, I always have to go to the toilet. So it's not surprising. The gut can also release gut hormones, which help the brain to understand that you're full, for example, after eating, or you need to eat, all that kind of things. There's also some microbiota metabolic derived products that can go in the circulatory system. And yes, so they're released by the microbiota itself. So if you have a good microbiota, it will release short-chain fatty acids, which will just help to decrease inflammation in general in your brain, and does also have other good benefits. The bad bacteria will release LPS, which is a very inflammatory molecule. So with increased inflammation, systemic inflammation, neuroinflammation in your brain. And that other pathway, it's the immune system. So I'm very interested in that one, because the gut can release also cytokines, means those cytokines, IL-1 beta, TNF-alpha, IL-6, going into the bloodstream, including inducing as well neuroinflammation. You already know that connection with the vagus nerve as well. So it could be a way of travel for the alpha-synuclein and going to the brainstem as well. And the vagus nerve also regulates some of the immune process as well. So all of that is still very unclear and still studied at the moment in animal models, but that's the very new and exciting area of research. Especially knowing that, your gut can impact your mood. So just think twice about what you eat, because, it can impact your gut microbiota and then your mood and, your way of having bad thoughts or everything like that. So I want to explain now what leaky gut syndrome is. First, in a healthy gut, you have normally in a healthy gut barrier. So this is the inside of your gut. You have the lumen of the gut. You have the gut microbiota here, good and bad. And normally, when you have a healthy gut microbiota, you have an epithelial cell barrier. So those cells will be connected together via tight junction. So that barrier prevents the pathogens from entering the body. When you have a stress event or toxin or food or medication, that epithelial cells barrier would start to be disrupted because they will lose their tight junction. Means that those components from the lumen can traverse into the lamina propria, which is another layer of the gut, and that can stimulate those inflammatory cells here, immune cells. Direct component as well from the bacteria can also cross there and go into the bloodstream, or also like directly bacteria can also traverse through into the bloodstream. This is very important because this link and that leaky gut syndrome inducing neuroinflammation has been linked now to many diseases as well. So neurological disease, so anxiety, depression, bipolar disorder, autism, schizophrenia, Parkinson disease, Alzheimer's disease, multiple sclerosis, and epilepsy. In Parkinson disease, I come back with the schema just so now you understand a bit better what's happening with the gut. So yeah, you have an inflamed gut, imbalance between the good and bad bacteria. You have those components going into the bloodstream, pro-inflammatory factor released by the immune cells going into the bloodstream. So enteric bacteria, food, also food allergen, pro-inflammatory factor, everything is in the bloodstream. Because systemic inflammation, mean inflammation, is in the bloodstream and everywhere in the body. That inflammation can cross the blood-brain barrier because it will be permeable, induce neuroinflammation. So that together with the olfactory bulb inflammation and also alpha-synuclein accumulation in the blood, in the brainstem, alpha-synuclein coming from the vagus nerve, all of those will link again to neurodegeneration of dopaminergic neurons. So now you really understand a bit better the link of the gut and the brain in Parkinson's disease, which actually makes sense because, yeah, a lot of patients with Parkinson's disease have that problem. So they have constipation and earlier on in the disease. What's very important is that there are anti-inflammatory approaches in diet, which is not surprising considering what I explained before with those inflammatory food and anti-inflammatory food. And especially those diets, such as the ketogenic diet. The ketogenic diet is a diet you want to reduce carbohydrates, almost no carbohydrates in diet, and increase the fat quantity and the protein intake. Intermittent fasting diet, it's just you have to fast for a few hours, then you can reinstall the meal. And Mediterranean diet, it's consumption of veggies, whole grains, a bit less of meat as well, olive oil. Yeah, all those diets would have been proven to reduce neuroinflammation, and there is a lot of clinical trial at the moment showing the impact on those diets on different neurological diseases. It's not new, it's already there. The problem is that you might not want to be on a diet if you have Parkinson's disease. So yeah, all those nice people have reunited together and they had that good idea, but so it was Louise, John, Sheila, Shakila, and Steph. They had that good idea that could we harness the benefits without being on a diet? So because again, maybe Parkinson's disease patients don't want to be on a certain diet. Maybe they don't want to lose more weight as well. And the good thing is like some of those diets, such as the ketogenic diet, would just release chemical compounds or dietary compounds already, or maybe it's just the dietary compounds, part of those diets that could just be helpful in decreasing neuroinflammation and also decreasing those disease. So here the idea is just to use those compounds, dietary compounds, to reduce the symptoms observed with Parkinson's disease by probably interfering with the gut and with inflammation. And probably other also damaging system that are occurring, we still don't know. So in Parkinson's disease, there is a limitation of the current rat models at the moment, because they're either toxin, means we induce dopaminergic cell death, but it's straight away almost all the dopaminergic cells, which is mostly what we observe at the end of the disease, but not at the prodromal phase or genetic models. And here we just want a more system-wide approach, including also inflammation in the disease. And previous lab member, so Connor Underwood was a postdoctoral researcher in our lab, and Grace at that time was another student, and she just came back 1 week ago, actually doing her PhD. So it's very nice to have her back here. And they were creating that model involving inflammation. So, and they did an excellent job because it works well. So the idea here, so this is the experimental design. I'm just going to explain what we do at the moment. It's still very preliminary data, so we just started that kind of studies. And I can't tell a bit more about how we induce inflammation or the type of dietary compounds we use because it's still very new. But I can just explain you the main idea. So we induce systemic inflammation for 3 weeks just in the water. Then we do a surgery after that 3 weeks where we induce a toxin in the striatum. The idea of inducing in the striatum means that it's only a progressive dopaminergic cell death. And we do that only one hemisphere, which means that one pole will be affected and the other pole won't be affected. So that's good because we can compare the motor skills of both poles. And we will do that drug treatment every day. We administer a compound for 8 weeks. 8 weeks. And during those 8 weeks, every week, we do behavior tests. So we analyze their motor skills by doing different motor tests, such as the cylinder test, the step test, the reaching, and the open field. Normally, those rats, when they have Parkinson, they are impaired in those tests. So here, the idea would be maybe it's just going to help them to improve their symptoms. And at the end of the study, we harvest the tissue and we collect the brain. The idea is to analyze the neuroinflammation in the brain and to see the extent of the lesion, to analyze the blood, analyze inflammatory markers in the blood, and analyze what's happening in the gut. So gut barrier integrity, inflammation, and maybe microbiota. That would be very interesting. So we haven't done that yet, just it's still very preliminary data as well, but it's some ideas. So preliminary results, so Grace and Connor, when they did that systemic inflammation in the rat model, so this is what you see in the colon, in the gut. On the left, it's a control rat, so they have a nice and healthy epithelial barrier, epithelial cells of the gut with nice crypts in the colon, which is very typical from the colon. Nice villi as well. When we see the Parkinsonian rats with systemic inflammation, that gut was totally different. And you could see that those crypts were not, the architecture was not great anymore. So it just disrupted crypts and also infiltration of inflammatory cells as well, which is what we wanted to observe. And then when they analyzed the cell death, the dopaminergic cell death in the brain. So we use a TH staining, so it's a marker of dopaminergic cells in the brain. And here's the substantia nigra. So this is what we have in a control rat. So we have the same dopaminergic cell number in both hemispheres. Parkinson's disease rat, it's because it's a lesion in the striatum, we have a re-reduction in the hemisphere, in the lesion hemisphere compared to the control hemisphere. And when we used systemic inflammation, we could see that that was even more decreased, so there were even more dopaminergic cell death. Means that inflammation probably was linked to higher cell death there as well. And now what I'm doing at the moment with that drug treatment, so still preliminary data again, but we saw that actually there was a decreased dopaminergic cell death with that drug treatment when the rats were treated daily. And we saw as well that the motor symptoms were improved with the drug treatment. Here it's a result with the step test, but I also observed that with another test. At that time, we were just doing the drug treatment until week 4, but here we're just going to extend until week 8 as well. And I just put a picture of Hannah because she's another student in our lab, and I think she's watching us tonight. So, hi, Hannah. And she's doing an amazing job at helping with all the behavior and all that project as well. So yeah, I hope she really enjoys it like we do. Yeah, and so at the moment we're in the phase that we were using one compound, but the idea would be to combine different compounds and to see the impact on the disease. But also we need to know the dose of that compound as well, because it's a new system we want to see, and maybe a higher dose of certain compounds would actually help to reduce gut inflammation. So for that, we need more studies and we need more funding. So the next step, I just want to show you a bit what we could do with neuroinflammation in the brain, just based on my previous studies as well, so you have an idea. And more can be even done as well. But so yeah, we could analyze just infiltration of T cells in the brain. So normally they're in the periphery; they don't infiltrate the brain parenchyma. So here, for example, it's an immunostaining using a marker CD3. Which will specific mark that's the T cells. So here they were infiltrating the brain parenchyma. So there was during a brain pathology as well. We can also analyze the brain by immunohistochemistry, the brain resident immune cells in the brain, such as the microglial cells, but also astrocytes. Those cells, they're normally present in the brain, so they don't infiltrate the brain. But when they're activated, they go from so in a healthy brain, they're not activated. So they have a small body shape, but with branch process, because they're scavenging the brain. But when they're activated, they start to be bigger and they lose their branches. And this is what's happening in brain pathology as well. So this is a good way of analyzing neuroinflammation, especially with those cells, because we know that they're involved in the pathological process. We can also do flow cytometry. So this is a very complicated technique, but that technique will analyze the expression of those cells, the expression of markers, if you want, on those cells, because we know that they will express different markers when they're activated. For example, microglial cells, when activated, they will express CD86, but also they go from CD45 intermediate to CD45 high. And you can see here, this is all the population of immune cells in the brain. And those ones represent the microglial cells in a healthy brain. So they are CD45 intermediate and CD11B. And they switch when there's a brain pathology towards those CD45 high. So that was very interesting to see that switch. And they also increase the marker of CD86. So this would be those population here in context of a brain pathology. So we could do that kind of analysis as well by using the drug treatment, and hopefully they would go back to the healthy stage and we wouldn't observe there anymore and they would go back to there. So it could be very interesting. We can also do quantitative PCR as well to analyze just the expression of some factors such as the pro-inflammatory markers within the brain. But we can also analyze those pro-inflammatory markers in the blood to see what's happening in systemic. For that, we can do ELISA, multiplex, also we can do flow cytometry, and we can analyze the antibody production in the blood too. So this is all measure of neuroinflammation. There are even more that we can do to see that blood-brain barrier, if it's permeable or not. Yeah, there are more to do. So I will just conclude my presentation now and just explain that we don't want to be too reductionist. We're just too reductionist by just think like it's only in the brain, you know. We know more that we need a system-wide approach. So by especially targeting the gut, importance of targeting inflammation during the lifespan by lifestyle, natural products, but also food. Anti-inflammatory compounds could be helpful to treat acute phase of diseases. Also need to take into account that there's that inter-individual variability. So everyone is different. So maybe some people would be more prone to inflammation where others not, you know. So maybe we will need to go towards a more personal approach for medicine in the future, especially considering the gut microbiota. Apparently everyone has a different one. And Parkinson in research, we need new animal models because at the moment it's too restrictive. Even the model we're doing, it's great, but we still don't have that part of the alpha-synuclein, so it's missing there. The gut-brain axis, we need more research. There's a lot going on there at the moment, especially around the microbiota, especially because it's involved in many neurological disease and psychiatric as well. The big picture here is to promote healthy lifestyle and sensitize the population about the impact on the mind, the body, and a healthy aging as a whole. And that was really interesting to show that as well, because I think that would be great if everyone can have an impact and increase all those healthy anti-inflammatory bubbles. So yeah, it's all on you. You have the power to act, you know, and to reduce those neurodegenerative and psychiatric disorders. So I would like to thank you, everyone, and thank you, the Neurological Foundation, for that opportunity to present. Thank you to the lab and all the collaborators as well. So yeah, this is the end. I'm just gonna stop sharing my screen now.
Oh, Elodie, look, thank you so much. That was absolutely fascinating. Thank you, everybody, for bearing with us through our technical glitches. Some really super questions already coming through down at through the Q&A. It is very much my great pleasure now to introduce you to Amy, who has come to rescue me. So, Elodie, thank you. That was absolutely fascinating. Thank you to everyone who's asked questions. I am going to hand over now to the very able Amy, whose technical glitches have been worked out, to talk us through the Q&As.
Awesome. Thank you, Elodie, and thank you, Kerry, too, and to all of you at home for bearing with us. Yeah, we've had some great questions come through on the chat, so we have a little bit of time left, so we'll try and get through as many of those as we can. But again, if we don't get to your question, I'm very sorry. We'll sort of try our best to maybe help answer that offline for you. But Elodie, one of the questions that has come through here, somebody has asked about an article that came out today, I believe. So you may not have come across it yet, but it's to do with mice, gluten, and inflammation. I don't know if that's something that you can comment on, or you can talk to maybe how that's impacting your research as well.
Yeah, actually, that's something I was interested in, and I wrote that as well in my review. I should have put that in one of those bubbles because, yeah, like, you know, there's different, you know, people are more sensitive or not to gluten, and those people who are celiac, of course, you know, that's their increased inflammation and everything. So yeah, we don't want them to have any contact with gluten because it's an allergy as well. But yeah, apparently even non-celiac people would have an increase of gut inflammation when they have gluten. So yes, but I think again that everyone are different, like I explained in my last slide. So maybe more people are, you know, more sensitized than others as well. But yeah, and I actually saw that. I didn't see that article, but I just saw that in one of the Otago release today. I don't know if it was linked to that article, but I was like, oh yeah, that's interesting. yeah. So yeah, it has been linked, and I think more research needs to be around that, because I know a lot of people who are like try to get rid of it from their diet, and they see a good improvement as well. So even if they're non-celiac, and yeah, definitely.
Yeah, cool.
Yeah.
Thank you. Now you did cover a few or a number of neurological disorders that are impacted, and somebody has asked if this could also be relevant to brain injury, so traumatic brain injury as well.
Oh well, yeah. That's funny. I saw that more in the, yeah, of course, in the other way. It's when you have a traumatic brain injury, you know, you have that increased inflammation in the brain, and that could be actually linked, you know, to other neuroinflammation type of disease, such as Parkinson. So this is one of the risk factors of Parkinson disease, for example, but it could be definitely linked to, yeah, other disease involved with neuroinflammation as well. Yeah, 100%.
Yeah. So another person has said that they're really interested in your thoughts on how vagus signaling could lead to neuroinflammation in addition to the blood-brain barrier permeable. Have I got that right? How could we target this pathway, the brain-gut axis, to reduce the rate of neurodegeneration?
So I just didn't understand you the first bit.
Sure.
Yeah, sorry. I'll just try to find it again.
Yeah, look, I'll pop it into the chat for you as well. But
that's good. Yeah.
Here we go. Yeah, interested in your thoughts on how vagus signaling could lead
vagus signaling. Yep.
Yeah.
Yep. Yeah, so interestingly, some people have some vasectomy. So we kind of cut the vagus nerve for different type of condition. I forgot which ones though, but apparently it has been linked as well to the decrease of Parkinson's disease. But of course, I wouldn't recommend to do that normally. I don't, probably it's probably like a lead type of
right.
Yeah, lead treatment. But yeah, and blood-brain barrier permeability, well, it's permeable when we have more inflammation going on. So I guess for me, my main idea is just to start straight away to decrease that inflammation, systemic inflammation, by acting already on the gut and try to reduce that systemic inflammation. I'm wondering if they must have, I try to think if they have they must have some drugs already working on that as well, like that could close that blood-brain barrier. I have to read more about that. Yeah.
We've had a few questions come through too, just around some of the diet that can help. Somebody commented that the ketogenic diet is included on the good side, but red meat is included on the bad side. Does red meat appear a lot in that ketogenic diet? What's the how do you separate those?
Yeah, I think that's a huge, it has been definitely linked to Parkinson's disease, and also like including evolving inflammation and things like that. So I would definitely, it's not totally canceling it, it's always that good balance. So of course you can eat red meat, just do it less often as well. I think for the ketogenic diet there are other sources of protein as well, so just maybe don't have red meat every day and just try to just use more chicken, that type of thing. Yeah.
Sorry, there's lots of questions coming in. It's great. Let's have a look here. It seems that symptoms can present themselves in midlife. What age group are you sort of talking about, and is it different between men and women?
No, I think, Parkinson's disease would typically start later on in life, but again, some people would develop it earlier on as well. So midlife, I think it's just it was more to show that those prodromal symptoms appear before the motor symptoms. But I think it's a bit different based on the person as well. But the idea here would be just really to focus on those clinical biomarkers before the motor symptoms appear. I think I'm not going to say that there's between 20 and 30 years old, or 30 or 40, because I think everyone are different, and again everyone are exposed to lifestyle factors in different ways as well. So I think it's just very different for every single person.
And do you have any other sort of hot tips for improving gut health?
But I think that was the whole purpose, with the inflammation in general, and Well, there's a
tips?
lot, and yeah, they're just, well, everything now apparently is linked, because I talk about the inflammation, but apparently everything now is linked with that, like kind of symbiotic living between us and the bacteria, between, you and the bacteria, and how everything would be linked to that balance of good and bad bacteria as well. So we just really want to please those good bacteria and say bye to the bad ones, so it's just really more about, and everything have had an impact on them. It's crazy to see what's out there. So we can act on them by eating healthy, yes, but like exercise as well would just, like direct those good bacteria towards a good balance as well. Everything is linked. That's crazy. Yeah. Just so yeah, exercise, eating. I would say like the eating part is the most important, and anti-inflammatory foods, and just be careful. Yeah.
Yeah. Just looking at the time here, I know we're running a little bit over, just due to a few technical issues earlier. But something I think that people are always interested to know in our community and among our supporters is what the benefits of sort of doing this research in Aotearoa, New Zealand are. Yeah.
Oh, well, I think, a lot of people are with, well, already just Parkinson disease. There are a lot of people with Parkinson disease in New Zealand. So, yeah, that's very important to, have, like I said, there is just a few treatment available. So, and it's only at the late stage of the disease. So if they knew that already earlier on, and even, it's not just doing that at the prodromal phase, even it would help at those late stage of disease as well. Exercising has been proven to help. Just be careful of what you eat as well. Just there's a lot of clinical trial at the moment just to help patient at that stage as well. And just I think it just needs to be more advertised, and I think definitely New Zealand would have a huge impact. Yeah.
Yeah. Right. Well, I think that's probably a good place to wrap up. Sorry to cut the Q&A a little bit short.
No worries.
But Elodie, thank you so much. We're so grateful, for what you do, but also for the time that you have given us this evening and the time that's gone into preparing for this evening. Yeah, we can't thank you enough. And also thank you to everyone who's joined us, and to our supporters and donors who enable us to keep doing what we're doing. So yeah, we really appreciate it. Just a reminder that there will be a video available in the coming days that'll be uploaded to our website, so you can watch this this webinar again, share it with your friends and family. But yeah, we'll leave it there. So thank you very much, and good night.
Thank you so much. Bye-bye.